You know where to find me. What? Are you just saying you can't see?
You want me to pull up the piano? You want me to pull up the piano? Or give you this chair and I'll pull this chair. You need one chair too.
Want to go with that or not?
I'll get you a note. I'm not sure either. All right.
Okay.
Are we live?
We are.
All right, welcome everyone to the second session of the 2015 Lakes REU Symposium here at Friday Harbor Labs. We're about halfway through. We have 8 more great talks ahead, and starting with Liz Allen from the College of William & Mary, and her mentor this summer was Sophie George.
Hi everyone, I'm Liz and I worked with Sophie this summer on the effect of freshwater influxes on Pisaster, ochraceus, larvae, and the Salish. So first, some background on why we're interested in this system. As we all know, global climate change is happening and it's causing global temperatures to steadily increase over time. This in turn is causing the Arctic ice to retreat at a pretty fast rate. This causes there to be more fresh water that's put into the oceans with nowhere to go. So this Arctic ice melt creates fresh water that enters river systems that actually come all the way down here because this area, the Salish Sea, is fed by over 14 rivers with the biggest freshwater influxes coming from the Fraser River. This graph right here has time on the X and salinity on the Y and shows the surface salinity levels here from Friday Harbor, from the Friday Harbor weather station from last summer. And you can see that every summer there are low salinity events happening throughout, with some being as low as 21 parts per thousand, where normal salinity you can see is at 31 parts per thousand. So this is a pretty big difference. The organism I worked with was the Pisaster ochraceus, which is found, its range is all along the western coast of North America, although this data is from the 1990s, and it's likely that this range has been significantly reduced since this data was taken due to epidemics of sea star wasting disease all along the west coast. Adult Pisasters spawn every summer from May to early June, and their development takes from 40 to 200 days in the laboratory and probably more than that in the natural environment. Since these low salinity events happen multiple times throughout the summer, the larvae must experience at least one low salinity event during their development. And so this begs the question of what effect does this low salinity water have on the sea star larvae, since we know they're going to be experiencing it at one point or another. So previous research on this topic has shown that there are morphological changes in the larvae of sea stars reared in low salinity versus normal 31 parts per thousand salinity. Here we see that the posterolateral arms here are significantly smaller in the low salinity larvae than in the control larvae. We also, previous research has also shown that Pisaster reared in low salinity show weird behaviors when they've been raised in low salinity, such as staying in low salinity water even though there's a halocline where they could move to higher salinity water below, even if there's food in the higher salinity water, they still will just persist in the low salinity, even though we know it's bad for them. So these morphological and behavioral changes mean that there must be protein expression and changes happening. And previous research has shown that there are protein expression changes happening. So my study investigated how low salinity affects the Pisaster mortality, morphological changes, developmental rates, and protein profiles. My hypotheses, based on these previous observations, were that low salinity reduces survival, that it creates morphological changes like we've seen before, that it slows their development, and therefore causes various up and down regulations of proteins in these larvae. My experimental design included 3 treatments. First, the controls at normal 30 parts per thousand salinity. We had a fluctuating salinity environment, which was held at 30 parts per thousand most of the time, but once a week for 24 hours, we would drop the salinity to 21 and then bring it back up. So they just got a burst of low salinity water and then were brought back to simulate the refluxes experienced here. But we also looked at an environment that was constantly at low salinity. This is important. This is important because it shows that the larvae can get stuck in the surface waters that have low salinity. Even though they're only here for a brief amount of time, the larvae aren't great swimmers, so they can get taken with it, which means they might experience low salinity for more than just a couple days, which is why it's important for us to know how important is this for their development if they are experiencing it for long periods of time. For larval rearing, we fed the larvae a mixed algal diet every 2 to 3 days of ochraceus, Dunaliella, and Rhodolomus. We also kept all these jars on a sea table to maintain temperature control, and we used a paddle design with a motor to keep the paddles moving to maintain motion within the water column to simulate the natural environment. So for my data, first, survival. Here on the y-axis, we have % of larvae surviving, and on the x, we have the treatment. This data is from the 34-day-old larvae, so most of the way through the experiment, and you can see that the survival was really high, and there were no significant differences between treatments, which shows that the low salinity isn't killing any of the larvae. It's not having an impact on their mortality in these closed systems. And it also shows that our methods of changing the water once a week also wasn't having an impact on the larvae's survival. We also looked at the growth and development. So this first graph on the left, you have the y-axis is the length of the larvae, and the x-axis is their age in days. And you can see that between these 3 treatments, there's no significant differences between their mean body length. So basically the larvae are all around the same size no matter what treatment they're in. However, if you look here, we look at the length of the posterior lateral arms, so these down here, and on the x we have age again. And you can see here they're all about the same, but here the low salinity is quite different from the control, meaning that their arms are significantly shorter. This is important because these arms are involved in swimming for the larvae. So here we have one of the larvae, and you can see it uses its arms to swim around and to orient itself in the water column. So it can help it get away from predators, help it swim out of the halopine, or help it orient itself so it can feed appropriately. And they're really cute. We also looked at the development. We used their coelomic pouches and whether or not they were connected as a proxy for how far in development these larvae were. So here on the right is a more developed larvae where you have the coelomic pouches connected up here, but over here you can see they haven't quite connected. So at 34 days old, there was a significant difference between the percentage of coelomic pouches connected in the sample size I took in the low salinity versus the normal salinity. The low salinity had significantly fewer coelomic pouch connections than the normal salinity and the fluctuating salinity environments. This shows that although they are the same size, these larvae are developmentally slowed down by this low salinity environment. This is important because if their development slowed, it probably means that it takes a longer time for them to reach metamorphosis, which means even though I saw no differences in mortality, we didn't have any predators. And if you're in the water column longer, it increases your likelihood that you're going to see predators. And so your mortality is probably higher. So before we did our protein analysis, we had to extract the protein. And we did this by first taking a sample of the larvae and lysing the cells with lysis buffer, which is what it looks like here. And then I performed a Bradford assay to determine the protein content within each sample of lysate, which is pictured here. The Bradford allowed us to figure out how much protein to load into the gels. And then we run all this lysate on gels like this, which would help us separate the proteins based on molecular weight. From previous research, we know what different size proteins, what their identifications are, so we actually can look at what protein is actually being upregulated or downregulated from the gel itself. So here's some data on the gels. For each of these, we have band intensity on the Y and the treatment on the X. Band intensity, it gives you an idea of how much protein there is, how much protein expression is happening. Here we talk about just various osmoregulatory proteins. And you can see that here, the control in the low salinity environment, there's no real changes in the amount of protein expression. But in the fluctuating salinity environment, you can see that this osmoregulatory protein is upregulated. Same case here, upregulation of the osmoregulatory protein. This is interesting because it's showing that the bursts of low salinity water are causing changes in how the larvae has to deal with the environment, because low salinity water changes how the ions are moving out and in of the cell, so it has to upregulate and downregulate different parts to handle that. But what's interesting is that the low salinity doesn't really change here, which might mean that the larvae has adjusted to the low salinity environment. And here we see downregulation, which could mean that that the larvae is actually working less hard because there's a stronger gradient in a low salinity environment than in a normal salinity environment. Down here are just pictures of the gels and what kind of data we look at. On the left in each of these is the ladder with we have known band sizes. And here you can see what we were looking at, 168, 112. We also looked at a mechanosensory protein. Mechanosensory proteins are what they sound like. They're mechanical proteins that are used for sensing. Beyond that, we don't really know what they do specifically in Pisaster. It might have to do with sensing different pressures or temperatures, but that we don't know for sure. But again, you can see the trend where fluctuating salinity has upregulated the protein, but low salinity, interestingly enough, is not upregulating and is again like the control, showing that maybe it's gotten used to the low salinity environment where the fluctuating salinity is still reacting to it. The implications of this research are that the slower development of the low salinity larvae could increase their chance of mortality prior to metamorphosis. Additionally, this data with the different banding sequences we have can be used for protein profiling of wild larvae just out here in the Salish Sea, to get an idea of what environment they've experienced. So basically, based on the banding sequences we get, we can tell whether a larvae has been exposed to a lot of low salinity during its development or not. Also, on a bigger scale, as global temperatures continue to rise, we can expect that these low salinity events in the area are going to increase in frequency, duration, and strength as the Arctic sea ice continues to melt at a faster and faster rate, dumping more water into these systems. For future work, I would like to do Western blots to help quantify changes in the mechanosensory and osmoregulatory protein expression, just to give us a better idea of how different is the protein expression and what's going on. I'd also like to do immunolabeling to determine where in the body these proteins are expressed. For instance, this could help us determine what is the mechanosensory protein really sensing? What is its function? Where does it function in the body of the larvae? And finally, I would love to look at how the Pisaster juveniles are impacted by the low salinity environment, and seeing whether growing up in a low salinity environment impacts them as an adult or fine once they metamorphose. I'd like to thank Sophie for mentoring me this summer and all of these people for helping me along the way.
We have time for questions. Bueller?
So you're casting their responses to low salinity as being a problem, that they're growing more slowly. Is there any way that growing more slowly would be an advantage to an animal that's in this situation?
From what I know, I don't think so, because I think they're most vulnerable when they're larvae. And especially based on the fact that their arms are reduced in size, that's definitely detrimental. There's really no way that having your arms growing slower is going to help you. So I think it's definitely detrimental. I can't think of how it might be a positive for them. I think it shows that they're spending their energy other ways. So maybe they're stressed and they're having to compensate and use their energy to help deal with the stressful environment, which means they're developing slower.
It's really beautiful work.
Thank you. Erika.
So you found that the low salinity ones had shorter arms and less developed coulombic pouches, and I'm wondering whether, because I don't know much about the larval arms, I've been thinking they're also responsible for feeding. So I guess my question for you is, do you think the not attached coulombic pouches are due to the low salinity, or a secondary result of the fact that they're not feeding as much, and so the coulombic pouches, in other words, has nothing to do with low salinity? Did you see any correlations with shorter arms and those stressed ones and less developed coulombic pouches? Because I think that would be the way to look at that. And
Yeah.
ones that happen to have the longer arms in that treatment had more connected.
Yeah, we didn't look at that directly, but actually we could pretty easily go back and look because we have photos.
I was going to say, I would think,
Yeah.
kind of, or if you know that that's number one and it was this,
Right, yeah, we
this,
could definitely look at that. But actually, the arms aren't used for feeding. The
Okay.
arms are just used for swimming, but that can kind of indirectly affect feeding because they need to orient themselves in the water column with the food patches to feed appropriately. So I think it can the arms definitely can affect their feeding, not quite directly. But yeah, like, it'd be interesting to look if those are related. And we could. Yeah.
All right, thanks a lot. Let's see. All right, our next talk is by Melissa Mayol of Muhlenberg College, and her mentor this summer was Erika Iyengar.
So good evening, everyone. All the talks that you've heard of as of right now have been all about the marine system, and mine's very different. This summer I spent basically my time in the forest collecting slugs and in the grassland collecting slugs, so I did a terrestrial system. And the two organisms in my system were the banana slug and the European slug. So the banana slug you see pictured in the top is characterized by this olive green color with black spots. It also has a distinctive keel running down its back. It's a native slug here on the island, and it's actually one of the largest slugs in the entire world as a terrestrial slug. And what's really interesting about it is that it has no similarly sized molluscan competitor in its evolutionary history. So there hasn't been much competition with another mollusk until the European slug was introduced. So the European slug, I'm going to call it the Arion here on forward. It was introduced from Western Europe. We think that the eggs were introduced either by like wood from construction or like plant material or something like that. So that came over, and Arion has two color morphs. And that in and of itself is very interesting because there's a lot of cool things happening with this population. So the two color morphs can be found in both the grassland and the forest area. So this brown color morph can be broken up into the grassland and the forest area. So when I say forest area, I'm referring to the Doug fir and the cedar firs that you can just find here on the fire trail. If you walk along in 5 o'clock in the morning, you'll find them because that's when it's moist. And then you have the black that are restricted just to the forest areas. And this is really interesting because we want to see if there's incipient speciation going on. Is there some type of difference between the grassland and the forest ones, or is there something going on between the black ones and the slugs found in the forest? And this played into my experimental setup because when I set up my experiment, I took this into consideration. And when I did my reps, I broke the Arion into black, I broke them into Arion Doug fir, and I broke them into Arion grassland. So my experimental setup, we did feeding preferences, observed there was competition going on. And the 4 choices that we used were because we saw the slugs feeding on these here on the fire trail and also through literature. So Sorfin and the lichen, Richter in one of his published theses, he said that the slugs consumed fern and lichen. He didn't cite these two in particular, but we decided to use these instead because we found them more commonly eating sword fern than bracken fern, which is what he cited in his paper. And we chose this species of lichen because this species of lichen he cited, we didn't really see them, and by we I mean Erika, because she's been coming to the island a lot more than I have my first time here. And then the third option that we presented them with was Pathfinder, and that's also from personal observations of Erika Iyengar and the paper that she is in the process of writing right now. She did research last summer to see what were the top choices that the slugs ate here on the island, and Pathfinder was the #1 choice. So that was the third choice that I presented them with. And then the fourth choice that I presented them with were store-bought mushrooms. And this is very different from everything else because they're store-bought, whereas everything else I had to go to the forest and collect. And the reason why I used a store-bought mushroom was because one of the papers that I read basically used as like a template for my research, used the store-bought mushrooms by Ryegrass wall. And so for me to draw comparisons from his experiment to mine, I had to kind of use store-bought mushrooms. Also, it's been super dry this summer, and for it to be, for you to have mushrooms, it has to be moist and it hasn't rained. So we're not going to get mushrooms, and we have seen mushroom, the slugs eating the mushrooms here. So, so that were the 4 choices that I presented my slugs with. Each slug was placed in a container, so they were all separated, 1 slug per container. I also had a control one going on where it was empty, so I could just use that to calculate desiccation and just things repeating of moisture to see what the weight change was, because that's how I calculated what they were eating. I would weigh them at the beginning and then weigh them at the end and see what the weight change was. So looking at my data, I got the banana slugs and I split them into 3 size categories, small, medium, large, just to see if there were any developmental differences going on. And if you look at my data, it doesn't seem that way across the board. All 3 size classes, they're eating basically the same amount, whether they're large, medium, or small. And this is really interesting because we think that the large ones that would weigh like 25 grams would be eating a lot more than the ones that weigh like 5 grams, but that didn't seem to be the case. And the #1 choice among all 3 size classes were the mushrooms. And also looking at the Arion, this was true as well. They all preferred the mushrooms compared to the Pathfinder, the sword fern, and the lichen. This wasn't really surprising that all 3 of them, so looking across, you can see that there aren't many differences across the 3 Arion, but what was the most surprising to us was that the grassland ate as much mushroom as they did. Because if you think about a grassland habitat, it's pretty dry, and they're not really exposed to mushrooms. Like, the ones in the forest really aren't exposed to mushrooms because it's dry, so like, it's even more rare for mushrooms to be found in the grassland area. So what that prompted the next question, the next step in my experiment was, okay, they all really like mushroom. Do they like mushroom because it's mushroom and the nutrients that they have, or do they like mushroom because of the moisture contained in it and everything else that the mushroom has to offer, like the texture? So what I did next was that I took the mushrooms and I dried them in an oven for 24 hours at 70 degrees Celsius, and I got lichen. And the reason why I chose lichen is because lichen is a combination of fungus and algae, so it's more likely to pick up moisture than a plant would. So I soaked that in RO water for 24 hours, and I presented the slugs basically in the same setup with the dirt, mushrooms on one side, and lichen on the other to see what they would prefer, and I would weigh them before and after. And the results were kind of interesting. Before they really liked the mushroom, they would just like hammer the mushroom, and that was their #1 choice. But now looking at this data, you can see that they all preferred lichen across the board, and that's made us think that, okay, maybe they do like the moisture more than they do like the taste, because being a terrestrial animal, desiccation is a really big thing that you have to worry about. So you have to get your moisture like through your food. So this kind of made sense to us when we found these data. So far the conclusions we can make is that they all have similar food preferences, whether it be banana and Arion that you're comparing, or all the subpopulations of the Arion. But then that prompted yet another question, because science kind of just snowballs like that. And we were like, okay, so climate change is something that is happening, and with that, moisture is changing and temperature can change as well. And since slugs are ectotherms, we're like, what effect will temperature have on these slugs? So what we did next is that we exposed the slugs to 3 different temperature treatments. 5 degrees, which represents like a springtime night, 10 degrees Celsius, which is a summertime night, and 20 degrees Celsius, which is a summer day. And so those were 3 different room temperatures. And then within that, once again, I put 1 slug, both brown, black, brown being both grassland, and milk fur and banana, small, medium, large, in these containers. I gave them 1 food option this time. I gave them spinach because from past data, they all love spinach, especially the Arion because they're a garden pest. So what I did, I took pictures before and after, and I used image analysis to see the area difference, to see what they would eat in the different temperatures. I also did this data with stinging nettle, but due to time restrictions, that data has not been touched. So what we were expecting from this was two different things. One, we expected on exothermic principles for them to eat okay here, like 4 degrees, not too stressful. 10 degrees that we'd expect them to eat the most because slugs are nocturnal, so you can expect them to be the most active at this time. But this is like a summertime night, so we expect them to be the most active at this time, and then for it to drop at 20. We'd expect 20 to be way too stressful for them and then not feed too much. And then Rodriguez Cabal, the person who was kind of like the template for my experiment, he did metabolic rates, and he used the same temperature ranges that we did. And he said with metabolic rate, it would increase with temperature. So we expected feeding to increase with temperature because just the metabolic rates going faster. But we did not find a difference that was significant. But this chart represents the Arion grassland. And this wasn't really surprising, because if you think about it, grasslands experience serious fluctuations throughout the day. It's pretty warm, cool and damp in the morning, and midday it's really hot, the sun's really beating down, and then once again it cools down at night. So you expect them to be okay and be able to handle these differences. And then for the second subset of the Arion, there was once again no significant difference. And this was more surprising. It didn't follow the trend that we set up before. But what was actually the most surprising for our data was that these large bananas did not, this was actually the opposite. We expected the 10 to be the highest and for everything else to be really different. But this is only an N of 5. I actually have a very large sample size that I haven't gotten to analyze yet. So maybe this trend will change. We are expecting and hoping it to, because we're kind of questioning as to what's happening here. Why is the 10 so low, whereas we expected it to be the highest because that's a nighttime temperature. So that brought into question, okay, why is it so low? And we had the humidity measured throughout our experiment, and we saw that the humidity is lowest at 10 degrees Celsius. So I made this question, okay, it ties kind of back into the beginning. Moisture, humidity, what's going on? And for future work, we want to see, are the bananas more sensitive to humidity? How important actually is humidity? And from the data that I just presented, it seems to be pretty important for their feeding rates. So my overall conclusions are that Arion feeding rate was not strongly affected by temperature, whether it be an Arion grassland, a dove fir, or the black slug. Also, the banana were not affected by temperature. And finally, that humidity can be more important than temperature, and that ties, as I said before, back into the whole mushroom thing, where humidity and moisture is really important for the slugs, where they do have to deal with that problem of desiccation and get moisture from whatever source they possibly can. So I would like to thank the Blink's Beacon program, Friday Harbor Labs, my lab partner Rebecca Potter for being awesome and helping me set up a whole bunch of tubs every night, and Rachel Mers for giving me property access. So thank you, and I will take any questions.
Once again, from the Twitterverse, do you think seasonal changes like this year's drought influence feeding preferences in slug?
I would say yes, just because, so with that, you have different things happening with the plants themselves. So if it's drier, you're gonna have different types of plants, I guess, growing at different times. So the slugs are gonna be affected by that. And they have a very specific diet. And when I say specific, yeah, they're general herbivores, but really they're eating pathfinder, which is limited to sunspots in the forest and things like that. So I would say yeah.
Any other questions?
How widespread are the European slugs?
They're pretty widespread. I mean, just the fact that they're found in more than one habitat, and then not only that, but they're super dense in the habitats that they are found in. So more so than the native. I don't know exactly the numbers of how widespread, but the forest is here, and then the grassland site is at least 10 miles.
I think he means biogeographically, how many states, countries.
Oh,
is that what you mean?
That was kind of what.
Oh.
Does Santa Cruz have European slugs?
I'm not sure.
No.
Alaska. Yeah. I would
So they're
Up
more
north, like
northwest.
British Columbia.
One more?
Yeah.
How did you control for your temperature and humidity? How did you,
So for my first experiment, which was really what humidity would affect, I had a control where I didn't have any slugs. I just had my 4 food choices. I would weigh them before and after just to see the desiccation or increase in moisture from that. And then I would like make a graph and base everything and adjust it off of that for each day.
I mean, like, did you stick it in the fridge?
Yeah, they were exposed to the same conditions as the other ones. They just didn't have slug in it.
All right, thanks a lot. All right, our next speaker is Rebecca Cotton from the University of Michigan. She also worked this summer with Erika Iyengar.
Hi everyone, thanks for being here tonight. I'm excited to be here, hope you all are too. Okay, this isn't working. Ditch that idea. So I was also looking at the slugs. We have, of course, the native banana slug, Ariolimax columbianus. That's over there on the left. Banana slug is a pretty well-known icon of the Pacific Northwest. I think it's a mascot of one of the schools. One of the California schools? Santa Cruz. Ah, there we go. Other people are more informed than I am. There is also the invasive European slug, Arion rufus. That's the guy over there who looks kind of like an alien. His intent is sticking up. Both of these are very large nocturnal creatures. They're herbivores with very broad diets. We see them feeding on things like stinging nettle and thistle. If I was a slug, I don't know if I'd want to eat those, but they apparently like them a lot. Don't know why. An interesting difference between the two is that, as Melissa mentioned, they are found in slightly different habitats. If you go into the forest at 5 o'clock in the morning, nice and early, you'll see both the bananas and the Arion slugs just cruising around. You can find them quite easily. However, if you go inland a bit and go into the grasslands, that's where you're going to find the Arion slugs. And the Arion slugs are very prevalent in the grasslands and in other disturbed sites, like people's gardens, stuff like that. They're so prevalent in people's gardens, in fact, that the eminent marine biologist, well known at the labs here, Eugene Kozloff, has said, if you have to hate anything, let it be this slug, a cruelly destructive pest, if ever there was one. Now that's a pretty harsh statement, but 1 year back in the 1900s, the Arion slugs completely decimated the strawberry population for the year. So if I didn't get any fresh strawberries that year, I would probably be disappointed as well. What can I say? That's not to say these slugs don't have any positive effects on ecosystems. There's been a lot of studies in forests that have shown that both of these slugs accelerate nutrient cycling and accelerate decomposition in the forests. Richter in 1979 published a paper showing that banana slugs, the native slugs, accelerate nutrient cycling, things like nitrogen, phosphorus, carbon, a lot of the other smaller, less well-known nutrients in forest soils. Steenhouse and Shu in 1996 looked at the Arion population and found that they will accelerate beach litter decomposition. And going back to the grasslands, I happen to know for a fact that there are a ton of slugs there. We have a resident slug queen in the back of the house, Miss Rachel Hers. She last year put on her property where there's a lot of tall grass. You see that there. Look out with binoculars. Early in the morning, you see slugs everywhere all over the grasslands. I've heard it said that in 15 minutes last year when it was actually wet and we weren't in this drought, she could easily go out and in 15 minutes collect over 70 slugs. That is a lot of slugs. Even now, in the middle of the drought, we went out very frequently, and in 30 minutes we get over 8 slugs, no problem. They are very prevalent on our grassland property. And so this really made me wonder, okay, we have these slugs. They've been shown to accelerate nutrient cycling in forest ecosystems, but they're a new species in the grasslands, right? The banana slugs only live in forests. These invasive Arion have been here less than 50 years. So what impact are they having in the grasslands? Are they still accelerating nutrient cycling like you would think? And so I decided to look at how Arion is impacting biomass production in the grasslands. And there's a couple different ways we think this might be happening. First of all, you have feces and mucus from the Arion slugs. Those are a source of nutrient addition. You would think that would have a positive impact on plant growth. But slugs are also herbivores. They might be eating the grass. They could be trampling young seedlings as they move over them, and maybe killing some of them that way. The mucus that they produce might also block grass stomata, breathing holes that grass is used for cellular respiration, photosynthesis, all that kind of thing. So there's a couple different positive and negative things that slugs may or may not be doing to the grassland ecosystems. So to control for these different positive and negative impacts, we had 3 different treatments. The first treatment had live slugs, and this was to account for all those various potential positive and negative impacts. The second treatment had feces only, and that was to say, let's just look at the positive impacts. If I just add feces, the source of nutrient addition, our plants are going to grow faster. And of course, for control, we had a no slug treatment. And so what we did is we took 100 ryegrass seeds in the little flower pots, and we put them in individual screen containers just to keep any slugs in that we put in and anything else out. You'll see around the edge here that there's a nice little black tarp, and that was provided as a shelter for the live slugs. We also had a wet sponge under there because, as Melissa talked about, these slugs will desiccate in the sun. They'll dry up and die. And they're nocturnal. They're not used to being out in the sun all the time. So we had that little sponge and skirt around all of our pots, just as a control. That little wet sponge could be impacting just how wet the soil is, local humidity, stuff like that. And so for our live slug treatments and our feces only treatment, we went out every other day and collected 80 slugs. Nice number. Half of those slugs went into the live slug treatment. We put 2 slugs into one of our nice window mesh containers with those grass seeds. Let them move around for 24 hours, poop, spread mucus, maybe eat grass, do whatever they're going to do. The second half of that 80 went into little individual Tupperwares. You can see over there, they have a nice little wet sponge in there to keep them happy. And they were again allowed to do their thing, poop, all that kind of thing. After 24 hours, we took the slugs out of the live slug treatment pots, and any feces that were in the Tupperware we put into our feces treatment containers. We wanted to get new slugs every other day so that we could be sure that the feces they had were from a natural diet. You know, if we feed our slugs lettuce, then the feces that they poop out have whatever nutrients lettuce has. But we were really interested in seeing what impact those slugs have when they have a natural diet and what's happening to that nutrient cycling. And then we got to sit back and watch the grass grow. Nice and early in the morning, because that's when the slugs are out. This is a picture of our final treatment that was set up. You can see our mesh cages out of the grid right there. Little inset to the right shows how our treatments were interspersed. The grasslands in the back were where we collected all of our slugs. We were very fortunate in that our slug collection site and our experimental site could be exactly the same place, so we knew that the slugs were getting exposed to the same environmental conditions. These boards up in front are what we use to shelter our feces treatment slugs, so the slugs in Tupperwares. And that way we would know that our live treatment slugs and our feces treatment slugs would be exposed to pretty much the same environmental conditions. At the end of the month, we took all our pots back into the lab and weighed above ground biomass. There's Melissa helping me there. We measured, that's the word I'm looking for, the length of the 5 longest grass blades, counted how many had survived the entire month, and then took wet and dry masses of the blades. We also looked at below ground biomass by sieving the soil that had all the roots in it, and then we again took wet and dry weights of the roots. And what we expected to find is that the feces treatments would have the most growth. The live slug treatments we thought might have the least growth because early on in our experiment we had witnessed herbivory. The grass had germinated maybe 3 or 4 days ago. We came back one day and quite a couple of those seedlings were mowed all the way down to the soil surface. The slugs had just gone and eaten them all up very happily. We didn't really observe much herbivory after that. We thought that initial herbivory, you'd think it would decrease plant growth. And this was not at all what happened. Surprisingly enough, we did not see any difference between our treatments, which was really rather odd. So on this first graph here, you're going to see grass length that was measuring our 5 longest blades. In the no-slug treatment, in the feces treatment, and in the live slug treatment, they did not differ significantly at all. The same is true for the total number of blades that survived the month. If you look at the weight of those grass blades, again, no significant difference. Looking at the weight, average weight per blade, maybe some blades are skinnier, some are fatter, maybe that's where their biomass difference is. Still nothing, no significant difference. The weight of the roots, below ground biomass, again, no significant difference. This is also true if you look at percent germination, if you look at the dry weights of grass blades and roots, you just really saw no effect. And so this was really, really surprising to us. Because remember, we saw that initial seedling herbivory, so we thought if nothing else, our live treatment pots would have less biomass. There's also other research that's been done in forest ecosystems showing that slugs do accelerate nutrient cycling, accelerate decomposition. They're taking nutrients from other spots and adding them back into the soil where plants can absorb them. And we don't know exactly what was going on with grasslands, but we didn't see biomass increasing. This was also surprising because a paper by Lauren and Whitlow published in 2012, which looked at the impact of Arion on the growth of Oregon grape, a perennial woody species. And this paper did find a biomass change when you added Arion into the woody grape. In low densities, Arion did increase biomass production of woody grape. Sorry, Oregon grape. That's the word I was looking for. At higher slug densities, Oregon grape growth was limited because of herbivory. And so to see something like Oregon grape, which again is perennial and woody, so it grows more slowly, be affected, and to have something like a ryegrass, this was an annual species that only gets 1 season of growth, so it's grown and then it dies, and for it not to be affected by having slugs present or even slug feces present was really, really shocking to us. One possible thing that the Oregon grape paper looked at is they found a slight trend in that having more slugs might impact other invertebrates that are in the area that may actually have more of an effect on feces or on nutrient cycling or herbivory. Obviously, our pots were in window mesh cages, so not many other herbivores were going to be getting into those. But we were really interested in looking at the direct effects of the Arions on grass growth. So, what can we say? That might be something to look into further. So back to Kozlov. He said, if you have to hate anything, let it be this slug. It would be really nice to tell you that even though this was a garden pest, they might at least be doing something beneficial for the native grassland growth. But this really doesn't seem to be the case. So there's a nice little Arion on this thistle. What can I say? They don't do much for our native grasses. I have to thank a lot of people for allowing me to be here at the Blinks NSF REU Beacon program and Friday Harbor Laboratories for just allowing me to be here this summer. It's been a wonderful summer. I've had a great time. Melissa Mayol, my brave research lab partner, suffered wasp stings, had Muhlenbergs, all kinds of fun things to help me out. Rachel Mers for very generously allowing me to use it for property analysis for help with grass identification. Thank you so much, and I'd be happy to answer any questions.
So, great job. I was just curious to see, did you observe the slugs feeding on your experiment?
I never saw them eating the grass. Usually by the time I went out to collect them, it was 9 o'clock in the morning and they were hiding in the shelters. Again, a couple days after we first saw germination, I went back and you could see that the grass had been mowed down to soil level. So you could definitely see that. After that point, it really doesn't look like they ate the grass. We did another experiment just snipping some adult grass that was out in Rachel's field and seeing if they ate any of that, and they didn't. So it doesn't look like they like to eat grass very much if they have a choice in the matter. But yeah. Yes?
What does the slug look like after having eaten from them? When you've got slugs, they're stuck in there, right?
So
and they're not eating the grass.
So sorry, I should have made this clear. What we did is every other day we put new slugs into our treatments. So we went out every other day, collected 80 new slugs.
So you had to open up your treatment.
Yes, yes. The treatments were rubber banded at the top, so you just open the rubber band, put the slugs in, we watered them.
Okay. So, I mean, it'd be interesting to do one with the slugs don't have shorts.
Don't have a choice.
Right? Because maybe you just found that slugs don't like these stuff for 2 days, right?
So they were, yeah,
So
it's a
that's,
possibility.
you know, that's.
You did that, Rebecca, that's the grass thing you just talked about.
Yes, so back to Bill. We did take adult grass leaves that we put in steps and put the slugs in with, a little container, had dirt, had the grass, had a slug. Left the slugs there for the same amount of time that they would be in our pots, and they did not eat.
Right, I'm questioning the time, right? So I'm just saying that, like the other study with that they love mushrooms and they don't love the other stuff, but if you take mushrooms away, then the other stuff comes up, right? So I'm just saying that an interesting direction would be, let's leave them there for a week and see if they'll eat that stuff, because then you hope now to see some better shows up during that kind of shorter time period.
It is possible they might have already eaten before you collected them, since you were collecting them in 2 days.
And I was hoping that they had. So I was hoping that they had eaten out of the field from the thistle, from the other plant species that aren't grass out there. Maybe from other species of grass, I'm not exactly sure. But whatever their natural diet was, I was really trying to look and see what their feces and stuff were doing to the grass growth. But yeah, it's a good point.
All right, thanks a lot.
What did you do with Arion when you were done?
In the refrigerator, they died.
Cool.
All right, our next talk is by Alexus Roberts of Purdue University, and her mentor this summer was Nick Gidmark.
My name is Alexus Roberts, and I spent this summer with my mentor Nick studying the biomechanics of the jaws and jaw muscles of Sculpin. So one of the easiest ways to define what we're all here studying at Friday Harbor is ecology. We're all looking at the interactions of organisms with other organisms and how they are competing against each other to obtain different resources. And when you have that competition, you are obviously going to have one organism that's better at obtaining a resource than another, thus competitively excluding the other organism. And to maintain diversity, you have to be able to figure out how these animals are separating out what they're going for and what resources they're obtaining and how. And so we call this niche partitioning. There's a variety of ways to study niche partitioning, including observing natural behavior, analyzing the diet of these organisms, biomechanical modeling, and I'm sure that you can all think of a few more. This summer, I wanted to look at the biomechanical modeling of the jaws and the jaw muscles in Sculpin to see how they might maintain their diversity in the same environments. Biomechanical modeling is basically quantifying data that's collected on the structure and the movement of organisms to understand their function and the functional relationships between the muscle anatomy and the skeletal anatomy. One of the most important pieces of information that you can get from this type of modeling is the amount of force that a system outputs. And in order to do this, you need to look at the skeletal lever system of an organism. The skeletal lever system is very similar to that of a crowbar, where you have an arm where force is being input and an arm where force is being output. So in the jaw system of fishes, the input, or the in lever, is the distance between the coronoid process, which is where the jaw muscles attach to the jaw and the jaw joint, and the out lever is the distance between the jaw joint and the tip of the jaw. So when you have that in lever, out lever, and you have that lever ratio, and you multiply it by the amount of force that's put into a system, you can get the amount of force that's output from the system. So when you have biomechanics of the jaw, you have specific bites that organisms have. And you can have an organism that bites fast or an organism that bites with a lot of force. And there are different traits that go into determining this characteristic bite. The first one is the skeletal morphology of that jaw. And with this, you're really looking at where the process is on the jaw. As that process gets closer to the tip of the jaw, you have a larger, stronger jaw. And as that process gets further from the tip of the jaw, you're going to have a smaller lever ratio, thus a faster jaw. The second thing that we have to look at is muscular morphology, which is the length of the muscle in that system when it's at the resting state. So if you have two organisms that have two different muscle lengths, let's say they have an equal mass, the one with the shorter muscle is going to have more fibers packed into that small space, but it takes a more absolute length change of that muscle to open the jaw a given amount. So that muscle is going to be stronger and slower. And then with the second muscle that has longer fibers, there are less fibers in that muscle, but it's going to take less absolute length change to open the jaw a given amount. So you're going to have a muscle that's faster, but weaker. The final thing that we have to look at when it comes to muscles and what goes into that characteristic bite is the force-length relationship that muscles have. Up until this point, muscles have been given a maximum bite force, just one value per individual. But what the force-length relationship tells us is that at instantaneous intermediate lengths, that muscle can give the maximum bite force. And when you have the muscle shortened from that intermediate length or extended from that intermediate length, the amount of force that it can output is much weaker. So here you can see that the muscle is about a half the length than it would be when it's resting, so it's shortened and it's giving off a very small amount of force. And then you can see as you get the muscle becoming more and more extended at those intermediate lengths, it gives off more force, and at the third length, it has the maximum force that it gives off, and once it's extended past that one, it gives off a weaker force. So what I wanted to do this summer is put all of these different traits of biomechanics into understanding how all of that works in the jaws of Sculpin. So I did this in a two-pronged approach. First, I looked at varying muscle lengths in vivo and in situ in these experimentations in 4 different species, just to look at how all of them use their muscle length curves. And then I also did an evolutionary inference with a group of species where I tried to figure out if the rectalis length and the lever ratio had any sort of evolutionary correlation. So due to time, I'm just going to focus on the evolutionary inference that I completed this summer. So Sculpin are a super diverse group of fishes. They're broken into 10 or 11 families. There are almost 800 species worldwide, and they're just super diverse, which makes them a great organism to study and just look at this biomechanical diversity in. The group of sculpins that I studied are all from the Burke Museum of Natural History and Culture, and they collected all of their specimens from around the San Juan Islands. So I looked at a group of 19 species, and as you can see in the pictures on the screen, they are totally diverse in their morphologies and just the way that they look. So I was able to get a really good understanding of their biomechanical diversity. So in order to complete this inference, I did a few things. First, I took the rectalis out of each specimen, and using nitric acid digestion, I was able to get an average length of the rectalis muscle. The rectalis muscle is one of the 3 jaw closing muscles in these Sculpin. And then I also took out half of the lower jaw, and after staining and clearing the jaws, I was able to get data on the in lever and the out lever and create that lever ratio. Once I had collected all of this data, I did a phylogenetic correction and I adjusted the data for body length. So when you phylogenetically correct something, you're correcting your data points for phylogeny. All of the species that I studied are related to a certain extent, and I needed to be able to study their evolutionary relationships independently of one another. So what we did was an independent contrast, and we gave one value of measurement to each node, and we also corrected the data for branch length. So after we did the phylogenetic correction, we linearly regressed all of our data, and before we even looked at our results, we figured that there were 3 different ways that the rectalis length and the lever ratio could be evolutionarily correlated. The first would be a positive correlation, where both the lever ratio and rectalis length are increasing over time. So here you would see that the jaw is getting stronger while the muscle is getting faster, or you would see the jaw getting faster as the muscle is getting stronger. So with this correlation, you see a trade-off between the muscle anatomy and the skeletal anatomy, and you also see that the system maintains a good use of force-link physiology. And the second type of correlation that was predicted is a negative correlation, where the jaw and the rectalis length are just moving opposite of each other. But when we put this into a system, you'd see that the jaw and the muscle are both getting stronger, or that they are both getting faster. So with this correlation, you have a common goal, and both the jaw and the muscle are working to do the same thing for that organism. The final correlation would be that there's no correlation at all. And so the jaw, the lever ratio, and the muscle length would be working independently of one another. So once I linearly regress the data that we collected, this is the graph that was generated. And here you can see that the data points don't really have a clear pattern. They don't seem to be going in a certain direction or way. And also notice that the p-value is well over 0.05. The adjusted R-squared is nowhere near 1. So with that, we can easily conclude that there's no correlation between the evolutionary shift in rectalis length and the evolutionary change in lever ratio. And so we see that these two things, these two traits, are evolving independently of one another, and that may seem somewhat insignificant, but it really means that these traits can do whatever is best, or they can evolve in whatever way is best for that species to survive and be successful. So now that I have that, I would like to look a little bit deeper into the in-lever and the out-lever themselves, and see if those traits have any sort of evolutionary correlations with head width or with the rectalis themselves. At this time, I would like to thank so many people that have helped me this summer. My wonderful mentor, Gidmark, a collaborator of mine, Stacey Gidmark, my awesome lab mates, Stacey Farina. Stacey Farina. My wonderful lab mates, Elska and Francesca and Catherine, for helping me with all of this. Everyone else, all my fish nerds in Lab 8, and everyone else at Friday Harbor for making this an awesome summer. Thank you.
We have time for questions.
Yes.
I don't understand the evolutionary connection.
Okay, so with that, all species are, at least the sculpins in general, are evolutionarily related to each other. And so to be able to look at those data points independently and try to see if there's a correlation between different traits, we had to correct for that. And so, go back. Okay, so let's just say that you have species 1, 2, and 3. And species 1 has a lever ratio of 6 and a muscle length of 30, and then species 2 has one of 4, a lever ratio of 4, and then a muscle length of 10. So if you take those numbers and you average them out, you're going to get a value for the ancestor between species 1 and 2. And so that would be at A, and their lever ratio would be 5, and the muscle length would be 20. And so we do that, and those are like the data points that we're plotting, and we're also looking at the branch length just to kind of adjust for those things so that the data points can be independent of one another.
One more, sure.
Did you try to look at the function or maybe an application of the differences in the lever ratios? So are these sculpins feeding the different meters or any different, like other different morphologies that may explain lever differences?
We haven't looked at that yet. That would definitely be something that we can look at to apply these things. Most of the sculpins are bottom dwelling. They're all just these bottom dwelling organisms with a bunch of spines on them. And they eat lots of other fish and shrimp and some crabs. So it just kind of depends on their overall morphology and what they're capable of eating with their jaw size. But what specifically is causing that level ratio difference would definitely be something to look into.
All right, thank you very much. All right, our next speaker is Elska Kaczmarek from the University of Miami, the one in Florida, and her mentor is Gidmark.
Hello. So this summer I looked at the force length relationship in the jaw closing muscles of king and king salmon. So in fishes, there's a huge variety of anatomies, morphologies, as well as behaviors. Functional morphology studies the connection between how a structure looks and how it functions, what it does for the animal. And each morphology is adapted to do a specific task very effectively. And so differences in behaviors between species are often reflected in the morphologies as well. And so you can see that reflected in the differing morphology of those two species. Most functional morphologists look at the skeletal system and not the muscular system, because the skeletal system is really easy to look at. It really has a lot to offer in terms of what's going on, but the muscular system also has a huge breadth of anatomical diversity, and it's integral to the functioning of the bones. Without them, the skeletal system wouldn't work the way it's supposed to. So by studying both of them, you can get really the most out of what's going on in that system. You can't really get a full picture without looking at them together. So I studied both the skeletal system and the muscular system in salmon, specifically king salmon and king salmon, which feed on different prey items. King salmon feed more heavily on plankton, so they'll swim through the water column with their mouths open and filter feed out plankton, but king salmon will bite down on small fish. They have to go swim and catch and bite those fish with strong muscles. Specifically, I looked at the adductor mandibulae, which is the jaw closing muscle. Salmon only have a single muscle to close their jaws. So that makes it really easy and simple to look at them feeding morphology. I looked at the force length relationship and how that characterizes their muscle. That's a relationship that characterizes all muscles. And what it says is that when a muscle is scrunched or when it's stretched out, it's not able to produce a strong force. But at an optimal muscle length called L naught, it can produce an optimum force called P naught. And that's directly related to the fish I'm looking at, because when the jaws are closed, those jaw muscles are being shortened. And when the jaws are wide open, muscles are being elongated and stretched out. And so at some mid-gait, they're able to produce an optimum force, P naught. To test these forces, we use this apparatus. So we needed the fish to be held in place when we contracted the jaws, and we used a clamp system to hold the fish down. We use a force gauge to attach to the lower jaw. So when that jaw, when the muscles contracted, it would pull on the force gauge and measure how much force is produced. That force gauge is also attached to a plate that can slide up and down. So we can change the gauge between each contraction and get all the intervals from full gauge range. There's a nerve bundle that controls the adductor mandibulae, and we would attach that to a wire, or the wire to the nerve, and then the wire also to an electric stim box, and that was used to contract the muscles when we wanted to. When we collected data, we'd start at a minimum gait, and we call that time 0, and then we wait 5 minutes and contract the muscle again. And then continue increasing the gape as we went until we reach max gape, and then go all the way back down and continue doing this to get multiple curves for an individual. Yeah, so they're all from the same individual, just going back and down over a few. And even though we waited 5 minutes between each contraction to try to prevent muscle fatigue, the muscle is still going to weaken over time. The animal is alive throughout the whole procedure because it's deeply anesthetized. And that's, again, supposed to elongate the lifespan of this muscle. But it's still going to weaken, and so over time, those forces decrease. And we quantified that to be able to see when the muscle started weakening, so that we can see, how it's influencing that curve, the forces that we're measuring. So we took the force decrease and plotted that against the time after that initial stimulation. And what we found is that for that x-intercept, which represents the time at which the muscle weakening began, It was between like 23 and 61 minutes after the initial stim, and for every specimen, that muscle weakening started after we completed that first series of data collections. So we knew that that first curve was not influenced by the muscle weakening. It was a good representation of the forces being produced. Then we scaled all of our data for maximum gait and maximum force production and plotted it for all of their individuals. The king salmon are in reddish colors and the pink salmon are in greens and blues. And for most of the king salmon, that curve is shifted over from where the pink salmon is. It has its maximum force production at a larger gait or muscle length. So our L0 is at a larger gait. And we also noticed that there might be a wider curve for the pink salmon. So we took all of our data points and created best fit curves for each of them and used that to measure exactly where L0 was, where optimal muscle length is, as well as what range of gaits could produce more than 75% of P0 for that individual. None of our comparisons were statistically significant, but I was only looking at 4 specimens of each species, really small sample size. And I think 0.06 is pretty good for that sample size. And with a larger sample size, I would expect to see some greater significance. But what we did find suggests that king salmon may have L0 at a larger gait than pink salmon, and that pink salmon may have a wider force length curve. So we know that pink salmon built to feed on plankton. We know that king salmon bite down on their prey, and we can see that we think that the force length curves are different in these ways. But what we want to know is how this could influence how they're eating and why it might be beneficial for them. And the way we thought about this is by looking at another relationship that describes muscles, which is the force-velocity relationship. So if you have a muscle contracting with a large amount of force, it's not going to, anyway, if you have it moving very quickly, it can't produce very much force. But if it's only moving slightly or moving not at all, moving little muscle movement, it can exert a whole lot of force. And it decays exponentially as you increase the velocity of that contraction. The other way to mess with that force production is by starting at a different gait. If you start at L0, you can get that P0 force production, which is the optimal muscle length. And if you start at any other gait than that optimal muscle length, the force you can produce is just no question can be less than P0. As well, if you move down the curve and you increase the velocity of your muscle contraction, you're also decreasing the amount of force that that muscle can produce. Fish can't bite down and catch their prey without moving their muscles and having a positive velocity, traction velocity. But what they can do evolutionarily is shift their force length curve to have their L0 at whatever, their optimal muscle length at whatever gait is best for them. So what we thought is that by having that curve shifted over towards a larger gait, the king salmon are able to eat larger prey items, and when they eat those larger prey items, have their muscles at an optimal length that they can produce a maximum amount of force and really get the most out of their muscles. Pink salmon, which don't have to bite down on their prey usually, they predominantly feed on plankton, and if they see a school of fish, they're not going to ignore it, they'll go after it, but really they're mostly eating plankton, so they don't have this kind of specialization. They have a broader curve that gives their muscles more range of functionality, and it's centered in the center, so it's not like designed to eat larger fish maybe, and maybe it helps with their keeping their mouths open for a long time when they're feeding. So in conclusion, we know that I need larger sample size to make my results significant, but what it suggests is that the muscle physiology reflects the behavioral differences that we see between the species. We see that the force length curves may be wider in pinks and shifted over in pinks, and this may be the result of other characteristics of the muscles that helps them feed more effectively. In the future, we want to increase that sample size and really get those results significant, and then maybe also explore the force-velocity relationship and see whether our predictions about what's causing this difference is really what's going on here. So I need to thank Gidmark for everything he's done, and Alexus and Catherine and Francesca for our Larvae, and I get to see them every single day. Carter for catching all of my fish for me, and Noah for a lot of the data analysis, which is computer technologies that I don't have. Yeah, is there any questions?
So the Twitterverse asks whether you were looking at saltwater or freshwater kypes, the kypes are those bizarre hooks in the jaws of salmon that they get when they start to move into freshwater.
So yeah, we caught all of our fish while I was here out fishing off the island, so it's all salt water. If that answers your question, is there anything else?
Any other questions?
Yes.
It's a weird one. So if being gaped open makes it optimal for it, how come fish don't swim with their mouth at their optimal gape all the time just so they're ready to go? Or like do sit and wait, predator fish that are going to jet out, just sit there with their mouth at the right opening?
I mean, their mouths are also used for other things, and it's really involved in breathing. So I think that having their mouths open would influence how they're able to really breathe and move their ventilation apparatus. I don't know if it's most optimal for breasting or for other muscle things, but it's for force production. So I can't really say.
Does the same fish repeatedly as it strikes its animal, its prey, like open its mouth to that, what you would predict is the right gape every single time?
So we're not working with fish that are alive and functioning on their own, right? So I don't really know. I haven't studied their actual behaviors. I've only just simplified it down to the actual force production in those muscles when we stimulate them.
Great, well, thank you very much. Our next talk is by Rose Dodgen of the University of California, Santa Barbara, and she was my student this summer. I was her mentor. Here she is.
So this summer, I looked at the group dynamics of the Maritime Earwig, Anisolabis maritima, and specifically how that's defined as their distribution within the environment. And I was looking at it with an eye to how this might be impacted by sexual selection, and specifically with a focus on finding what the mating system structure for this organism might be. So group dynamics and dispersion patterns of an organism can tell us a lot about its mating system. There are three types of distribution patterns: random, uniform, and clumped. And a random pattern, like this one, is difficult to interpret. It can be impacted by a lot of different factors. It's hard to tell what a mating system might be for this type of pattern. However, uniform, like this type of distribution, suggests territoriality, like you might see in male frogs. And clumped distributions suggest two possible types of mating systems: harems, which are groups of females that males compete for access to, and leks, which are groups of males that form and pose or compete for female attention. Females will come visit them. Now, harems are a kind of mating system that's seen in another common beach arthropod, the California beach flea, or Megalocestia californiana, shown here. But because female maritime earwigs are pretty aggressive, it doesn't seem to be too likely that this is the mating system for this organism. On the other hand, previous research has shown that male earwigs are willing to cohabitate in small-scale experimental trials. So the idea of a male grouping system like lekking seems to be a more feasible possibility. Now, lekking is usually observed in birds, like the sage grouse that you see here, but it has been described in several orders of insects, though not many earwigs. So my study species, Anisolabis maritima, is found all over the world. It lives on sandy beaches under driftwood, so it's not really a marine species, but we can squeak it. The species is notably sexually dimorphic. Males have curved forceps, and females have straight forceps. Females are also generally slightly larger than males and more aggressive. They won't hesitate to use their forceps to attack and kill intruders or competitors into their areas, and they use this aggression to defend their nests, especially during the summer, from other females who might seek to cannibalize their eggs. Males, on the other hand, tend to have non-lethal interactions. All earwigs have curved, all male earwigs, that is, have curved forceps, but asymmetry is unique to this species. And males will use their curved forceps to pinch and squeeze the abdomens of other males to either gauge size or try to flip the other male over and establish dominance. So there's been a lot of previous research in this area, and it gave us a few, when we were coming into this, it gave us a few things to keep in mind when we were thinking about what the mating structure of the system might be. First of all, we've seen that larger males are more competitively dominant, but between smaller size-matched males, forceps asymmetry lends a competitive advantage. So if we're looking at groups of competing males, it's something to keep in mind. We've also seen that in a small-scale research trial with 3 earwigs, males are willing to cohabitate, but females are not. And we've seen a strong sexual preference for large individuals in both males and females. So when we came to this project, we had a few hypotheses about what structures we might find. Among groups of just males, we predicted that they were more likely to be either randomly distributed or forming groups based on their non-lethal interactions and their willingness to cohabitate in previous research on small scales. Females, on the other hand, because they're so aggressive and because they maintain nest territories, we predicted were more likely to be uniformly distributed within the environment. Mixed sex groups, we predicted would both the male and female patterns we predicted lend themselves to the idea of lucking, with females establishing territories and males forming clumps and being visited. However, we thought it might also be the case that because females are so aggressive, it would prevent males from forming groups and produce a uniform habitat or uniform distribution where female aggression prevents grouping and maintaining harmony throughout the environment. So our earwigs were taken from Albury Island. The largest and most accessible population is at False Bay. Back in the lab, we marked them with a B tag. It's a small circular tag with a color and a number. You glue it directly behind the head on the thorax, and that gives them an ID to distinguish them during the trial. It's been shown not to affect their behavior. After the trials, they were frozen, and then we measured them. The width of their 6th abdominal segment was used as an index for body size. It's a standard procedure that's been used in previous literature. And when we were looking at distributions after our trials, we compared them to our Poisson distribution, which is a model distribution that illustrates what a random distribution would look like under experimental circumstances. And anything that differed significantly from the Poisson distribution was analyzed with an index of dispersion to demonstrate what types of patterns were forming that caused it to be significantly different. So when we were looking at distribution, we had these Erwin trials, and these were designed to look at large-scale group dynamics and distribution patterns. They consisted of 18 individuals, either single-sex groups of all males and all females, or mixed-sex groups of 9 males and 9 females. And they had 9 square red plastic shelters. Earwigs can't see red, so these would appear dark to them, and this was meant to mimic their driftwood habitat while still allowing us to see them during trials. And what we can see here on the bottom, this is what the distribution patterns might look like if we saw them in one of our arena trials: a random distribution, a uniform territorial distribution, or a clumped harem or left distribution. So what did we find? Groups of just males were randomly distributed at 6 hours, but over time aggregated and formed clumps at 12 hours. And I should mention, the way these trials were set up, late in the afternoon, the 6-hour check was late at night. Because these earwigs are nocturnal, 6-hour checks would have been during their peak activity, whereas the 12-hour checks were early the next morning when earwigs would have settled into positions that they would likely maintain for the entire day. So we can see males are random, but then they form groups and settle into groups for the remainder of the day. We think this might be the case because female aggression limits the amount of territory that males can occupy when they're under driftwood, and males who are willing to group with other males increase the space and resource availability to themselves. If we look at females, we see that they're uniformly distributed during the peak activities, which suggests that they are maintaining aggressive predicted, but over time that ceases to be, and they randomize. And it seems likely that without a nest or another resource to protect, aggressively defending against all the other individuals in the arena becomes unnecessary energy. So as the other bugs in the arena become more familiar, you seek to defend your area so aggressively. When we looked at these sex groups, we saw that at 6 hours they were uniform, hours they were clumped. And at first, I at least thought that this might lend itself to the idea of lucking with females forming territories and the males forming clumps to be visited. But if you look at the mixed sex distribution, If you look at the individual sexes distribution within this mixed sex pattern, there doesn't seem to be too much support for that. But it's true that females are uniformly distributed at 6 hours, and it seems to be the case that their aggression and their forming of uniform pattern among themselves is what's causing the uniform pattern to appear all over within the entire arena. They're aggressive to both males and females, and they are establishing distinct territories. However, like in the single-sex trials, over time, they become randomly distributed. They are still reducing their aggressive interactions over time, and they aren't changing their behavior in response to males. On the other hand, males in mixed-sex environments are randomly distributed at both 6 and 12 hours. And what that tells us is that they're not forming clumps like they normally would on their own. And this could be because they're off chasing females, it could be because they're being chased off by females, but whatever the reason, it's clear that female presence and aggression is what's altering the behavior of males and therefore changing and controlling the distribution in a mixed-sex environment throughout the night. And the fact that the overall distribution of the mixed-sex group is clumped at 12 hours could be for a couple of reasons. It could be the case that because there are fewer females, there's a less incidence of female conflict, which allows females to be more tolerant of other individuals being near them and allows groups to form despite their aggressive tendencies. And it could be the case that males are forming groups, but because there's a low density of males in a large arena, we're not seeing that pattern when we look at just the male distribution within this data. And that's something that could be looked at further with more arena trials of a higher density of males and a lower density of females. We also conducted a series of smaller scale tub trials, and these were just to look at cohabitation preferences as a proxy for sexual selection and mating preference among 3 earwigs, 2 different sized individuals of one sex and 1 individual of the opposite sex, so 2 a large male, small male, and female, for example. And here's what we found. Overall, there is a huge preference for smaller mates. We have twice as many males choosing to cohabitate with a small female and twice as many females choosing to cohabitate with a small male. And although this is a common pattern, we think the reasoning behind it for each sex might be slightly different. But when a male is choosing between 2 small females, or excuse me, when a male is choosing between 2 females, a larger female might be a more attractive option. They're likely to be better nest defenders, they're likely to be more gravid, and in previous studies, we've seen that a male will choose to cohabitate with a larger confined female. The note there is that they're confined. A confined female can't kill you. In these environments, a large female can and will kill you. And so choosing to cohabitate with a smaller female or male is probably better for your health. If females are aggressive and competitive, so it's likely that smaller males present less of a threat to them, and they're more willing to let them cohabitate within their shelters. But what's really interesting is if we look at the size differences between individuals who can who tend to cohabitate with larger individuals of the opposite sex. If we look at a male choosing between 2 females, we see that the average male cohabitating with a large female is much larger than the average male cohabitating with a small female. It seems to be the case that only really large males can defend themselves against large females, and therefore are able to cohabitate with the most desirable mates. And this presents a possibility of assortative mating with large individuals tending to group together. On the other hand, if you look at females, the females who are cohabitating with the large males are much smaller than the females who are cohabitating with the small males. And because females prefer small males, it seems to be the case that large males are able to force small females to cohabitate with them against their will, especially in our arenas where small shelters allow small females to be cornered, and therefore small females can be forced to cohabitate with a large male even if they prefer a smaller male. So overall, we've seen that males are willing to cohabitate and form groups, females are not. Females are very aggressive, and that aggression controls the mixed sex environment within the Erwig habitat. Now, traditional leks weren't seen, and given the female tendency to hold territory, the thought of a female visiting a group of opposing males as in a traditional lek doesn't seem super likely. But because males are so likely to form groups, another type of male grouping system might be at work, and that's something we could look at more in the future. And then when we're looking at sexual selection and the mate choice between the earwigs, we see that smaller individuals are preferred, either because they're less scary or they're easier to control. But large males have the advantage of being able to gain access to the most desirable mates in large females, and that presents the possibility of assortative mating, which is another thing we could look at in the future with more small-scale trials of distinct size classes of individuals. I'd like to thank the Blinks NSF REU program and my wonderful mentor, Iyengar, and all these wonderful people here for giving me the chance to do this research. I'm happy to take questions.
Do these animals mate more than once in their lifetime, or are they just once in
Mating season is throughout the summer, and they live for about 3 years in the lab, or at least that's the record that I read. So it's likely that they mate several times over their lifetime.
That would make sense about the whole males going for smaller females, because if they know that they're not going to get killed, they want to preserve for the nest.
For a chance to mate again, yeah. Other questions? Erika.
See, I know you put them in the arenas and you didn't sit there for 4 hours and it got to be 1 a.m. in the morning, but I'm just wondering, does the amount of aggressive interactions taper off over time?
Yeah, that's something we talked about looking at in the future. We don't really know, and without something like a stop motion camera to gather footage throughout the night, we won't really be able to tell, but it's something we've talked about, setting up a camera and filming these interactions throughout the night to see if that is the case, because I think that is what's happening here. Melissa.
So to record your data, you weren't recording the actual interactions, you like the snap?
No, we had a chart set up, and we would go in at 6 hours and 12 hours and mark the locations of all the units in the trial on the chart, and then, yeah, that was our data.
Take one more question.
Yes?
Can you tell anything from the distribution?
When you're collecting them in the field, they run away real quick when you get the log up, so not really. Females tend to stay on their nests even when you get the log up, so you might be able to see that their nests are distributed over a pretty wide area. They don't tend to have 2 nests close together, but because they disperse so quickly when you move their environment, it's hard to tell.
So you could actually maybe measure the distance.
You could potentially measure distances between nests, and looking at nests was one of the things we talked about doing this summer, but you only have 8 weeks to do all this, so.
All right, thanks so much.
All right, our next speaker is from University of Washington, Jasmine Fuerte Stone, and her mentor this summer, her mentors were Ted Mayol and Andrew Clark.
So this summer I was assessing muscle function in the feeding apparatus of the Pacific hagfish of Dodgen. Pictured here, this is a bird's eye view looking down on the Pacific hagfish in its resting coil position. Hagfish are jawless, elongate invertebrate fishes. They're strictly marine, found from 15 meters to 5,000 meters below the surface. They're opportunistic scavengers and can often be found in animal carcasses. Despite their being jawless, they are able to bite with surprising force in order to tear the flesh away from these carcasses. One way they're able to do that is by nodding, as seen in this video. Looking at that again a little more closely, there are several attempts to tear away flesh unsuccessfully, and then it knocks its entire body in order to get enough force to actually pull away. So that biting cycle that you saw is a cyclic protraction and retraction of a tooth plate. Here is a side view and a bottom view of a hagfish protracting and then retracting its tooth plate, which is this white area. And this is done in a book-like fashion, so it opens and then opens up again and then goes back into its mouth in reverse. Up close, here's the tooth plate. There are a couple rows of keratinous teeth shown more closely. This is the protraction and retraction is powered by a feeding apparatus, which is located by the mouth. This is belly side up of a hagfish. Here's its mouth. This is the feeding apparatus. It is made up of cartilage and muscle. So in blue is cartilage. This is top side or bottom side and top view. Here's the tooth plate that we saw protracted before. So muscles in the feeding apparatus. The heart component is where the cartilage is,
but the soft component is where there is mostly just muscles. In the hard component, there's superficial protractor muscle, which lies on top of the feeding apparatus, as well as several heads of a deep protractor muscle. And the soft component is comprised of a sphincter muscle, which is long and tubular, a retractor muscle, and a perpendicular muscle. The retractor muscle is actually inserted into the sphincter muscle and attaches onto the tooth plate. And that is where it's thought to have most of the force for retraction and biting. So previous studies have looked at the function of feeding muscles in Atlantic catfish using EMGs in live feeding. And what was found was that deep protractor muscles were the only muscles activated during protraction, whereas all three muscles in the soft component of the feeding apparatus were activated during retraction. I was studying the Pacific hagfish, which has a slightly different feeding apparatus in both size and shape of the muscles, and our main question was, do Pacific hagfish feeding muscles function like those in Atlantic hagfish despite these differences? Unfortunately, we were unable to do live EMGs with feeding because our hagfish would not eat all summer. So we, so we instead decided to apply voltage to mimic action potentials from neurons and contract the muscles, and recently euthanized animals. So we had a simulator which would provide a squirt pulse of voltage to the muscles using electrodes made out of standard field electrodes and some cano wire that would stiffen and allow us to direct our pulses. And we mainly simulated these highlighted areas up here for protraction and down in the soft area for retraction. So what we found was that Atlantic and Pacific hagfish do indeed function in much the same way. The protractor muscle has four heads, as I said before, the medial, which are these two middle ones, and the lateral deep protractor, which is on the outside. Only the medial deep protractor muscle would cause protrusion when stimulated, whereas the deep protractor muscles do not. And then all three muscles in the feeding in the soft component of the feeding apparatus would cause retraction when stimulated, which was what was seen with EMGs. Previous studies have also looked at the kinematics in Atlantic and Pacific hagfish, again in live feeding. Here is a chart looking at times of the protraction and retraction cycle in Atlantic hagfish in red and Pacific hagfish in blue. As can be seen, a lot of the timing was very similar between the two. Here's protraction at about 300 milliseconds and retraction for about 600 milliseconds for both. When we try to replicate this using simulation, we found that while protraction at 400 milliseconds was very similar, our retraction was only 150 milliseconds, which was surprisingly short. But upon looking back at our videos, we saw that there was a time where between when we would stimulate protraction and then stimulate retraction, where the feeding where the tooth plate would actually fold back into the mouth without the electrodes on any of the muscles, which can be seen in this video. So there is protraction, electrodes come away, and it slightly folds back in. And this was relatively slow and actually makes up about 250 milliseconds of time, bringing our total retraction time much closer, and could indicate that retraction is partially passive.
Can you play that one more time?
Yes. So protraction, pull away the electrodes, and then it pulls back in. So there's a proposed strain for the feeding apparatus of Atlantic hagfish, and it says that it is like a cylinder, and that maintains constant volume no matter if it's protracted or retracted. So when protracted, it would become short and fat, and when during retraction, it would become long and thin. And we want to know again if this hagfish strain in this manner as well. In order to do this, we took measurements of the length and width during protraction and retraction. Length was then converted into longitudinal strain, and width was converted into transverse strain. Strain is the change in length divided by, or width, divided by your initial length or width, and our initial length was what we took when it was retracted.
And it's important to note that this is a unitless and normalized number that helps describe the overall change. So what we found was that the longitudinal strain was negative, meaning that it got shorter, which is what was predicted, and transverse strain was positive, meaning that it got wider. Again, this is what was seen in the modeling of the Atlantic hagfish. So we also had a question of whether or not excising the feeding apparatus had any impact on kinematics and strain. So the feeding apparatus is connected to the rest of the body, mostly by soft connective tissues, so just fibrous connections, which are easily cut away. And so we were wondering if these had any impact on their ability to protract and retract. So here's a video showing protraction again, but one more time. So differences in kinematics, we felt time-wise that there was significant, or there's a great difference when pulled out of the rest of the body. It took about twice the time for protraction. However, the time for retraction was about the same. Strain-wise, longitudinally, the excised feeding apparatus changed more than the in situ or when it was within the body. However, for transverse strain, there was more change when it was in the body than when it was outside the body. Overall, however, total shape change is about the same. So this could indicate that deformation remains constant, and even though there might be some constrictions placed upon it, it is not enough. Probably isn't enough to change the protraction and retraction, but that's something that we'll have to look into more. So our initial question was, do Pacific hagfish feeding muscles function like those in Atlantic hagfish? And we saw that yes, they do. Comparing the EMG work to our simulation work, deep protractor muscles cause protraction, the soft component causes retraction, which is what was expected. Do Pacific hagfish muscles strain in a similar manner? We saw that that cylindrical model did hold up with what we saw in our experiments. And does excising the feeding apparatus change kinematics and strain? And while it does change a little bit of strain lengthwise and widthwise, overall it seems that total deformation does not change. So why are we even working with these? One reason is to understand the evolution of chordate feeding mechanisms. So hagfish are between jawed vertebrates and basal chordates, and they represent this transition from soft ciliary feeding to forceful jawed feeding. And this transition step and understanding it in this understudied animal can help give a more wholesome explanation of how feeding mechanisms work. There's also application in soft robotics. So soft robotics are robots that do not have any hard components and will be able to perform tasks that may be hard for something with a hard component because they're able to manipulate their shape and maneuver more easily depending on terrain. So things like search and rescue, surgeries, especially in like veins, maybe guts. And they're also able to provide force that could perform tasks like injecting or retracting, as seen in feeding. I'd like to thank the Blues NSF REU Beacon program, our advisor Vic, my two PIs, Andrew Clark and Ted Winnow, my lab partners, Christina Perez and Austin Haney for all their work, Adam Summers for helping us get specimen and housing our animals, and Nick Gidmark for providing MS-222 so we can utilize our Are there any questions?
There's time for questions.
Yeah. Any questions? Yes.
So hagfish are famous for their slime.
Yes.
I'm just wondering how much, as you're exercising this, etc., the slime might be getting, I mean, I'm sure you were careful, but how much, is it possible to avoid slime getting all over them and perhaps interfering with you, stimulating them, measuring the stuff, etc.?
So, yeah, the slime is glycoprotein and will not expand if it's not in water. So because we took them out of the water before we started dissecting, the slime comes out as kind of like a really thick mucus, and you can just kind of wipe it off and get rid of it. Yeah.
So you have this like cylindrical model for the muscle strength. Are there any other models to how that muscle would change?
Not that I'm aware of.
Questions?
Yes.
That whole apparatus reminds me a lot of the Ragil apparatus of mollusks, the way it's operating. Had you thought about that analogy?
It's been brought up, but we didn't look into it. No.
All right, thank you very much. All right, our final speaker of the day is Catherine Korn of Cornell University, and her mentor was Adam Summers.
All right, hi. So as you all know, my name is Catherine. I studied here as Adam's REU student this summer, and we wanted to know about scaling and its relationship with burial in the English soul, herbivorous vegetables. So scaling is all about how life is different at different sizes. So if you've seen Ant-Man, you know that things look very differently when you are tiny. But your whole environment changes, you know. Sand, when you're small versus when you're big, can be, you know, a little mountain for everything that looks like a tiny little grain as it is here. And big is not just longer. So if we start thinking in 2D, not just 1D length, surface area scales with length squared, and then 3D, mass, volume, scales with length cubed. And so, you know, to sort of visualize that, if you have a 5-centimeter fish who grows to be a 30-centimeter fish, that's a 6 times size difference. What if this fish is a territorial fish? Does the territory he occupies that no other fish may enter without facing his wrath increase by 6 times? Because the territory is a area, it's a 2D metric, and so it scales with surface area. So it goes up 36 times. So a 30-centimeter flatfish will occupy 36 times the territory that a 5-centimeter flatfish would take up. But we didn't look at territorialism in flatfishes. We looked at how they get into the territory that they're in. So we studied burial in particulate solids. And particulate solids, so sand, granular media, are really special because they have both fluid properties and solid properties. So if you take some sand in your hand and you open it up, it will flow out of your hand like a fluid. But if I were to drop you on that sand, it would not be like falling into a pool. It would be like falling onto a solid. And that has a lot of really cool implications for how organisms can move when they're moving with it and when they're moving through it and around it. And so, you know, with burial, there's terrestrial burial by moles and snakes and rodents, but it's really hard to get a solid object into a semi-solid substrate and back out both fast and reversibly. And so with fish, there are 3 major players in our equations. We have the organism. So, as you may guess, because my talk is about scaling, the length of the fish makes a difference. And you also have to take into account the way that the organism moves. So that's its frequency. And then the environment around it. So the sand, the size of the sand can also be scaled. You can have big sand, you can have little sand. You're going to move differently in those two. And then the density of the sand. And most importantly, something that we often forget in our own lives is water. So the media surrounding our basic, like, benthic environment. With a fish, you have to take into account movement of the water and the way that the water flows, and it can take a lot to get up the energy to move and overcome that inertia.
So flatfishes are really cool to study as barriers because they don't really look like something that buries at first glance. This large flat surface area, if you drop that on some sand, it's not a wedge. It's not going to nicely sink in. It's like a, when you drop a plate on the ground. It doesn't go well. But they're actually really good at this. These are fishes that are almost designed to live in the media below them. One of their eyes rotates around to the other side of their head, which is the really characteristic flatfish look. And though it looks like they're dorso-ventrally compressed, they're actually laterally compressed and dorsal. And so this is what, oh, right, thank you. So this is what flatfish burial looks like. And so as you can see, there's a bobbing motion of the head and the tail. And the fins are also undulating. And the sand is fluidized by the fish up over and around to cover the top of the fish. And so that was a 17-second video. That burial event, actually took 1.3 seconds. This has been slowed down a lot. So what we want to know is whether time to burial and the frequency of that head and tail bobbing motion, so we'll call it an undulation, scales with fish length, if length and frequency affect the percent covered, and if this is dependent on scaling of the fish size or scaling of the particle size. So the first experiment that we did was to change the fish size. So we took 15 fish from 5 centimeters to 30 centimeters, and we had them bury 5 times each in the same size sediment. And we looked at 3 things: that frequency, the amount of time it takes them to bury, and then percent coverage. So these fishes often bury all the way down, leaving only their eyes exposed, but sometimes they don't, and that can be really informative. So frequency declines with length. The bigger the fish, the slower it undulates. The smaller the fish, the faster it undulates. We clocked some of the little 5 centimeter fishes going 14 hertz, whereas the bigger fishes went like 3. And the bigger fishes also take longer to bury, which sort of goes with this concept of slower undulations taking a little bit longer. And, it's a bigger fish, it has a larger surface area and a larger mass to move. With percent coverage, we saw no relationship. So no matter how slowly or how long it took them to bury, they still buried enough to the same degree. But is that really all? So we changed the grain size. Once again, we can either have animal size or grain size change. And so we wanted to look at relative grain size to the fish. So we kept it consistent and used little fishes with with 5 different grain sizes, spanning 125 to 700 microns. There we go. 5 fish, 5 sediment sizes. So our baseline hypothesis was that grain size would not affect burial metrics, or our null hypothesis. Maybe all of these fishes are going to bury equally well in all the different grain sizes, and actually the only difference is based on the size of the animal. Alternatively, since larger grain sizes tended to, or larger relative grain size was with smaller grain size and larger fish, or relative to the fish. So the bigger fish and the smaller grain sizes was a slower undulation frequency. We expect a larger grain size relative to our small fish to be correlated with faster burial and faster undulation. But grain size doesn't affect frequency. And this relationship holds true for time to burial too. So grain size, undulation rate, time, there's no trend there. But with the bigger grain sizes, you have a lot less coverage. The percent coverage here was much, much higher for the same size fish burying in the smaller grain.
And so what we get out of this, not only are bigger fish slower, but it's due to the effect of scaling on the fish size. And though larger grain size yields less coverage, there's no change in fish undulation or in their time. All right, thank you.
Time for questions.
Do you think that the slower burial in a large fish is because the fish is trying to do something different, or that it's the physics of the fluid and all those material properties that are basically forcing a larger fish to bury slower?
I mean, to a certain point, if it's trying to vary, you know, the ultimate goal would be the same thing. I would at least definitely consider when you are a 30-centimeter fish, a lot fewer things can eat you than when you're a 5-centimeter fish. And so, you know, there may be, I guess, a little bit less need to bury 100% and be completely covered. But, Paolo?
Yeah, so I'm from the fish swimming class.
Yeah.
We're asking a question that relates to fish swimming.
Go for it.
So, in fish swimming, if you look at any given speed, for example, burst swimming, then what you would expect is that frequency would balance the size of the stuff. You have some swimming. However, if you're using muscles like the one that you're using to swim. So I don't know if you have to fit everybody can have it. It'd be interesting to look at how the slope of size versus frequency relates to the slope that you get for something like burst swimming. And if you don't have it for flatfish, you can just use the general equation for fish and feed. Because it's not exactly the same as swimming, but any difference may reveal something interesting about it.
Absolutely. Yeah, thank you. Yeah.
I have the same kind of thought as Nick did, and I'm wondering, so I assume you did not have a predator in the lab when you were doing this? And it might be hard enough. But like it may be hard to do with your big fish to get a predator that's scary enough, but with a mid-sized fish, I wonder that you're already seeing that they're going slower. I wonder if you put a very big visible like predator or picture of it or simulated what, you know, that all of a sudden if you saw them going, oh shoot, and they do it a lot faster and suddenly seem to look at the same. Because I can imagine being a big fish and just being lazy and not sitting around. It's not a big deal. Does that make sense?
It does. I mean, they definitely, you know, if they did not bury within a certain, you know, I give them a couple minutes and then it's like, all right, you can expect me bugging you now. I'm going to come with my
Right, and then would they suddenly bury?
Not always, but sometimes.
Yeah.
So that was pretty uniform across. The big fish could expect that, the little fish could expect that. But my hand is not quite a fish's mouth.
So I was thinking like eagle. Yeah, slap them in and see what happens.
That would be beautiful. Catch me an eagle. Yeah.
When you see it in the field, though, it seems like they have some ability to control the amount that they're going to put, energy, effort they're going to put into burying themselves. So when I was looking at the no correlation of how much they covered themselves, it was like, well, for each individual, it looked like there wasn't a huge range of variation. That most of the variation came from different individuals.
Are you referencing the grain size differences?
No.
Oh, okay.
The amount of covering themselves. With the different size fishes.
This one.
Yeah.
Hang on, can you say it again?
Well,
I just
it looks
want to.
like most of the, it looks like a lot of the variation where you don't have a correlation is because each individual fish has its own little range in which it buries itself, and that seems to be pretty consistent. But when you see them in the field and you start watching, you start harassing them, they sometimes bury themselves a little bit, and then once they make up their mind to it, they can bury themselves pretty good. So there might be something else to consider.
That's definitely something we could look at. Yeah.
Any more questions?
Is there any difference in the amount of time they stay buried?
That was not something we tested, but it's definitely something we could. Maybe. I'm curious about whether other parameters would influence that. Like some things that we don't have or that we weren't looking at, presence of food nearby. I would unnerve it.
You did say there's a difference in range, or like territory they inhabit.
When testing the different sand sizes, sand grain sizes, do you know the densities of the particles? Because I was wondering whether getting more or less coverage with different particles had to do with the density and the amount of time it took for the sand to fall down and sort of pushed it over.
Right. And I imagine that the density would also change with the grain size. Like you'll have other, or because just from
All right, great, thanks. All right, I just want to say a couple quick words, a couple thank yous to people like Scott Sweeney, who's behind the scenes here. And I'd also like to thank Adam, who's the PI of the REU grant, and for thanking him personally for allowing me to run the program now for 3 straight years. But most of all, I want to just thank everyone here, all the students, all the mentors, for all the hard work that they put in. I think it was quite evident throughout the 4-hour session tonight. So thank you very much. And this concludes,